Hello, everyone. I'm Greg Smith, the Curriculum Coordinator for the BioNetwork Bioprocessing Center. And welcome to today's BioForum entitled Current Topics in Bioethics. Our BioForum is scheduled to run from 10 a.m. until approximately 11 a.m. Eastern Standard Time. I'm excited to announce that our panelist today is Sheila Mikhail, founder and managing member of Life Science Law. In just a moment, I'll turn it over to Sheila, and we will begin today's BioForum. We'd like you to feel free to ask questions by typing them in the chat box located on the right side of the screen. I'll be keeping a record of each question that is submitted, and we'll have a time at the end of the presentation to address all of your questions. Be sure to send chats to all participants. This will ensure that we don't miss your question. Please mark your calendar for our next scheduled BioForum entitled Blended Learning on May 7th, 2013 at 10 a.m. Registration for this event is free and will be available online at ncbionetwork.org. We'd like to know who we have in our audience today, and at the conclusion of today's webinar, a poll will ask you just a few short questions. Please select the options that best describe you. Also, we want to have an accurate record of your participation in today's BioForum. Please let us know if you are sharing a screen by typing the total number of participants at your location in the appropriate question. If you're the only person viewing at your screen, please just type the number 1. Thank you. If at any time during today's presentation you experience choppy audio caused by a slower internet connection, try closing the audio broadcast panel, then click Communicate, and then Join Audio Broadcast to reconnect. We would like to remind our audience that if you log on to our website at ncbionetwork.org/IET, you can take advantage of our free interactive e-learning tools that we have created. These cover a variety of topics, and we're certain that you will find some of these that you can use. If you participate in social networking, we're also on Facebook, and you may keep abreast of the topics in the news, future events, and other interesting articles by liking us. Simply go to facebook.com/BioNetwork. Thank you. We're excited to have each of you with us, meeting with us today, and at this time, I will turn it over to Sheila Mikhail, our presenter.
Good morning, everyone. Before I get started, I'd like to thank Greg Smith for all of his efforts in coordinating this presentation. I'd also like to thank the BioNetwork for giving me this opportunity to present today. This morning, we are going to spend some time discussing bioethics, which is a very interesting subject. Hopefully, I pulled together some slides that will allow you to explore some of the more tantalizing aspects of some of the current issues facing society today. The field of bioethics merges various aspects of several different disciplines, including biology, medicine, politics, and the law. The role of human subjects in the investigation of therapeutics gives rise to several concerns within the realm of bioethics, which concerns increase over time. This presentation will provide an overview of some of those topics. Sorry, it seems like my slides just jumped a little bit more than they should have. The overview today, we're going to talk. I'm so sorry, I'm having a little bit of difficulty. Hang on real quick. There we go. There's 4 topics we're going to talk today. First is the ownership of biological samples isolated from patients. Second is the patentability of isolated gene sequences. Third is the safety of study participants in human clinical trials as it relates to the process of obtaining and ensuring informed consent. And finally, we're going to look at the use of drugs that have not been yet approved by the FDA for those patients who have no other treatment options available to them. The first topic is, who is the rightful owner of biological samples that are isolated from patients and used for medical research? To best illustrate the issues surrounding ownership of biological samples isolated from patients, I want to start off with a story about a remarkable woman named Henrietta Lacks. Henrietta was a poor tobacco farmer who lived in Virginia during the 1950s. One day, she felt a knot inside of her. She assumed that she was pregnant. Her local doctor confirmed that she was correct, but after giving birth to her fifth child, Joseph, Henrietta started bleeding abnormally and profusely. Her local doctor referred her to Johns Hopkins. Now, at the time, Johns Hopkins was Henrietta's only choice for a hospital. It was the only one in the proximity that treated black patients. Howard Jones, her doctor, examined Henrietta and the lump in her cervix. He cut off a small part of the tumor and sent it to the pathology lab. Soon thereafter, they discovered that she had malignant cervical cancer. As was common for the times, Lacks was treated with radium tube inserts, which were sewn in place. After a few days, the tubes were removed and she was released from Johns Hopkins. She had to return for X-ray treatment as follow-up. During her radiation treatment, 2 samples of Henrietta's cervix were removed, a healthy part and a cancerous part. Both were removed without her permission. Shortly thereafter, at the age of 31, Henrietta died from cancer that had metastasized throughout her body. But the cells that she left behind would become one of the great important tools in medicine. There's been a lot written about Henrietta, including several books recently, and Oprah produced a production about her experience. Henrietta's cells that she left behind were very unique. Researchers discovered that they did something that they had never seen before. That is, they could keep alive and grow. Before Henrietta's cells were discovered, cell cultures from other cells would only survive for a few days. And that was a problem because scientists would spend more time trying to keep the cells alive than performing actual research on the cells. But the cells from Henrietta's tumor sample behaved differently than the others. The researchers were able to isolate one specific cell, multiply, and then start a cell line. And the sample was named HELA, and that was after the initial letters of Henrietta Lacks' name. These were the first human cells grown in a lab that were immortal. What do I mean by immortal? Well, that is, they didn't die after a few cell divisions. As immortal cells, they could be used for conducting many experiments, and these experiments could be conducted by different researchers across the globe at the same time using the same cells. This created an explosion in the feasibility of conducting scientific research. By 1952, HELA cells were traveling by planes, trains, and automobiles, and even pack mule all over the world. Her cells eventually ended up even in space. Within a few years, companies began selling HELA cells and the media to grow them. Today, the HELA cell line is one of the most important tools in medicine. Henrietta's cells have been bought and sold by the billions, creating a multi-billion-dollar business. Yet she and her family received no financial compensation. In fact, Henrietta's family had no knowledge about the commercial success of the HELA cells and the contribution they made to science. Henrietta's family only learned about the HELA cell story after the family in the mid-1970s beginning calls from researchers who wanted blood samples from them to learn the family's genetics, that is, the eye colors, their hair colors, and genetic connections. The family wondered why they were getting these calls, and it was later from these calls that they learned about the removal of Henrietta's cells. For those who are unfamiliar with the role of cells in scientific research, this slide gives a little bit of a primer. HELA cells are an important type of cell culture, and cell culture is the ability to maintain and grow cells of multicellular organisms outside the body in specifically designed containers under precise conditions. Cell culture is important as it serves as a model for basic cell biology. Cell culture enables many studies to be done, including the effects of drugs, the process of aging, toxicity studies, production of vaccines, production of biological drugs, among other things. The HELA cells were instrumental in many scientific discoveries, and some are presented on this slide. Perhaps you can identify some of them. The first job that HELA cells was used for was in testing the polio vaccine in 1952. A lab accident using the HELA cells led to the discovery that normal human cells have 46 chromosomes, and that was obviously important for many scientific discoveries that followed. HELA cells were important in cloning, and here we see a picture of Dolly the cloned sheep. They are also key to the development of stem cells and in vitro fertilization. At least 5 Nobel Prizes have been awarded for research involving HELA cells, and it is estimated that 50 million tons of HELA cells have been grown since 1951. So indeed, HELA cells have made very significant contributions. Could Henrietta's story occur today? Could the cells be taken from a patient and used without their knowledge for scientific research and perhaps even for profit? If we were to look at the informed consent that Henrietta signed back in the 1950s and compare it to the one that she might sign today as a patient at Johns Hopkins, it appears to be very likely. So what are the laws governing the use of tissues taken from a person? Generally, it is illegal to sell tissue for transplantation or medical treatment. Thus, the sale of a kidney for money is not allowed. It is, however, legal to sell tissue for research, education, or art. In fact, Ted Slavin, who contacted hepatitis B in the 1950s during a treatment for hemophilia, sold his blood for significant amounts of money. His blood contained exceptionally high levels of hepatitis B antibodies, which were commercially valuable. And he sold his serum in the 1970s to laboratories and companies for over $10 per milliliter. But there really aren't any laws governing human tissue research. There are laws protecting privacy, as we all know when we go to the doctor's office and have to sign the HIPAA form. The HIPAA rules govern the use and disclosure of health records. There are also some laws that protect basic safety of human subjects, most notably the Common Rule. The Common Rule requires independent bodies such as institutional review boards, otherwise known as IRBs, to review the safety of clinical trial protocols and outlines the guidelines for informed consents. However, the Common Rule does not apply to tissues unless the sample is individually identifiable. In such case, the patient is required to give his or her consent for the use of that sample. That means, however, that research is permitted without a patient's consent if the tissue is anonymous and minimal identifying information has been given, or if the samples are left over, and in some of the other situations listed on the slide. As the Common Rule is not specifically written for tissue use, much is left for interpretation. Let's look at a few case studies applying the principles that we learned in the last few slides. The first case study is Washington University versus Catalona. Dr. William Catalona, preeminent surgeon and expert on prostate cancer, was conducting research at Washington University. During his research, he collected tens of thousands of tissue samples, and he received the appropriate patient consent. Those samples were stored at Washington University. Eventually, Dr. Catalona moved his medical practice to Northwestern University. When he did so, he contacted all his former patients and asked for their permission to transfer their tissue samples to Northwestern University. Approximately 6,000 men signed the new consent forms, allowing Dr. Catalona to move the samples and continue the research they originally donated the tissues for. Washington University filed a lawsuit to block Dr. Catalona from moving the samples. Washington University asserted that the men gave a gift to the university and not to Dr. Catalona, and therefore the men could not dictate what happened to their tissue samples. The research participants objected. The men argued that the informed consent documents they signed, agreeing to participate in the research, demonstrated that they retained ownership rights over their own little flesh and blood. The forms promised the men to withdraw from the research at any time and even promised them the right to have the sample destroyed upon their request. So if they had the right to destroy their sample, the men argued, clearly someone else didn't own it. They asked to have the samples moved to Northwestern University so Dr. Catalona could continue his research. But Washington University asserted that it had the right to use the men's samples as it wished in its sole discretion. In spite of the language in the informed consent documents, allowing the participants to withdraw at any time and for any reason. The university argued that the patients had given a gift, a donation to the university, and they could not take it back. The Eighth Circuit Court of Appeals, in affirming the decision of the trial court in favor of Washington University, held that the men who participated had given their tissue to the university as a gift, and they could not get it back, nor could they have it sent to another research institution. However, the court indicated that men retained the right to stop participating in the research by declining to answer any additional questions. They could refuse to donate additional tissue, and they could disallow the use of their tissue in future research. So in conclusion, what we've learned from this case study is once you donate tissue, you have no control over who possesses them. In our second case study, we're going to look at the use by Arizona State University, or ASU, of an Indians tribe DNA samples. Members of the Tiny Huvasavi Indian tribe have given DNA samples to ASU researchers starting in 1990 in the hope that they might provide some genetic clues to the tribe's devastating rate of diabetes. But they subsequently learned that the blood samples had been used to study many other things other than diabetes, including mental illness and theories of the tribe's geographical origins that had contradicted their traditional stories. ASU, as a research institution that receives federal funds, was required to receive informed consents from the subjects, and that informed consent required that the subjects understood the risk and benefits before they participated. But the protections were designed primarily for research that carried physical risks to the subjects, like experimental drug trials or surgery. When it comes to mining DNA, the rules and the risks are much murkier. After spending $1.7 million fighting lawsuits by the tribe members, the university's Board of Regents agreed to pay $700,000 to 41 of the tribe's members, return the blood samples, and provide other forms of assistance to the impoverished tribe. The settlement appears to be the first payment to individuals who said their DNA was misused, but it left behind a lot of lingering issues. In our third case study, in our final case study, we're going to look at a case called Moore versus Regents of the University of California. In this case, John Moore underwent treatment for hairy cell leukemia at the UCLA Medical Center under the supervision of a Dr. David W. Gouldy. As part of the treatment, Moore's spleen was removed, and he signed a consent form allowing the surgeon to dispose of any severed tissues or member by cremation. Moore's cancer, however, was later developed into a cell line that was commercialized by a biotech company from which Gouldy had received compensation. The cell line, called the Mo cell line, was worth billions. The California Supreme Court ruled that Moore had no right to share in any of the profits realized from the commercialization of anything developed from his discarded body parts. So even though Moore had provided the tissue that developed into this billion-dollar cell line, he had no right to receive any profits associated with it. So that concludes our discussion on the first topic. And the next topic, I want to look at how do we ensure the safety of study participants in human clinical trials. As we all know, the overarching objective of clinical research is to develop generalizable knowledge to improve the health and/or to increase the understanding of human biology. By placing some people at risk of harm for the good of others, clinical research has the potential for exploitation of human subjects. Ethical requirements for clinical research aim to minimize the possibility of exploitation by ensuring that research subjects are not merely used, but are treated with respect while they contribute to the social good. The ethics of experimentation on humans was derived from the lessons of history and some of the abuses that resulted to vulnerable populations. Let's look at a few of these on this slide and the impact they had on bioethics. First, the Nazi experiments. As we know from our history lessons, Nazi human experimentations was a series of medical experiments on large numbers of prisoners, mostly Jews, from across Europe. were conducted by the Nazi German regime in its concentration camps, mainly in the early 1940s during World War II. They included a variety of experiments, including bone, muscle, and nerve transplantation experiments, head injury experiments, freezing experiments, malaria experiments, mustard gas experiments, sterilization experiments, among many other heinous other experiments. After the war, these crimes were tried at what became known as the doctor's trial, and the revulsion of the abuses penetrated led to the development of the Nuremberg Code of Medical Ethics. The next history lesson that impacted bioethics development is Holmesburg's prison. This prison was part of the City of Philadelphia prison system. It was built in 1896 and continued in use until 1995. Between 1965 and 1966, Dr. Albert Kligman, a dermatologist at the prison, exposed approximately 75 prisoners at the prison to high doses of dioxin. This was, as many of you might recall, the main poisonous ingredient in Agent Orange. Dow Chemical paid Dr. Klingman $10,000 to conduct the experiments on the toxicity effects of the Vietnam War-era chemical. Dr. Klingman exposed prisoners to a dosage of 468 times greater than that in the Dow Chemical protocol for the experiments. And obviously, it resulted in many injuries to the patients and many undesired health effects. The next history lesson is custody. The Tuskegee syphilis study is cited as arguably the most infamous biomedical research study in U.S. history. The syphilis experiment was an infamous clinical study conducted in Tuskegee, Alabama by the U.S. Public Health Service to study the natural regression of untreated syphilis in rural black men who thought that they were receiving free health care from the U.S. government. This involved withholding of penicillin, which was known at that point in time to successfully treat syphilis, from these patients and denying them alternative treatment that was available to others. In fact, in 1966, Henry Beecher wrote an article in the New England Journal which identified 22 examples in which patients had never had the risk satisfactorily explained to them, and it seems obvious that hundreds others did not know that they were the subjects of an experiment, although grave consequences could have, and in some instances actually were, suffered. Among Beecher's 22 examples, including withholding antibodies from men with rheumatic fever, injecting live cancer cells into nursing home patients, and transplanting melanoma from a daughter to a mother who subsequently died. For the past 50 years, the main sources of guidance on ethical conduct of ethical clinical research has been the Nuremberg Code, the Declaration of Helsinki, the Belmont Report, International Ethical Guidelines for Biomedical Research involving human subjects, and similar documents. As stated previously, the Nuremberg Code was part of the judicial decision condemning the atrocities of the Nazi physicians, and so focused on the need for consent and a favorable risk-benefit ratio for experimentation, but it makes no mention of fair subject selection or independent review. The Declaration of Helsinki was developed to remedy such gaps in the Nuremberg Code, especially as it related to physicians conducting research with patients, and so focuses more on the favorable risk-benefit ratio and independent review. The Declaration of Helsinki also emphasizes a distinction between therapeutic and non-therapeutic research that is rejected or not noted by other documents. The Belmont Report was meant to provide broad principles that could be used to generate specific rules and regulations in response to U.S. research scandals, such as Tuskegee. It focuses on informed consent, favorable risk-benefit ratio, and the need to ensure that vulnerable patients are not targeted for risky research. The Council for International Organizations of Medical Sciences, or otherwise known as CIOMS, guidelines were intended to apply the Declaration of Helsinki in developing countries. particularly for large-scale trials of vaccines and drugs. Good clinical practice, otherwise known as GCP, is an international quality standard that's provided by the International Conference on Harmonization, or ICH. ICH is an international body that defines standards which governments can translate into regulations for conducting clinical trials involving human subjects. Good clinical practice guidelines include protection of human rights as a subject in clinical trials. It also provides assurance of the safety and efficacy of newly developed compounds. These include standards on how clinical trials should be conducted, define the roles and responsibilities of clinical trial sponsors, that is the biotech or pharmaceutical company that has developed the drug, clinical research investigators, and monitors. The common rule is a rule of ethics regarding biomedical and behavioral research involving human subjects in the U.S. These regulations govern institutional review boards for oversight of human research. The common rule is the baseline standard of ethics by which any government-funded research in the U.S. is held. Nearly all academic institutions hold their researchers to these statements of rights regardless of funding. There are also various federal regulations governing the ethics of clinical research. So what makes research involving human subjects ethical? Most researchers, bioethicists, and institutional review members would answer the informed consent. The informed consent ensures that individuals have the opportunity to decide whether they want to participate in research or continue participation, and whether it is compatible with their goals, values, and interests. For those of us who have participated in a clinical trial, we would have seen an informed consent. For those of us who haven't, what is an informed consent? Well, an informed consent is a legal document that should have 4 elements contained in it. One is disclosure, and that is a statement of the research to be conducted, its purpose and procedures, its foreseeable risk and discomfort to the patient, any benefits to the subject that's participating or to others, if there are any alternatives, identification of those alternatives, treatment or compensation for injury. If something were to go wrong in the clinical trial, will the subject be reimbursed for any medical treatment that he or she might seek? Participation is voluntary. That's a statement that at any point in time the subject could withdraw from the clinical trial. These are the elements that you would see in any legal document that would be presented to you before you participate in the clinical trial. But in order for an informed consent to be effective, there has to be an understanding by the patient of what he or she is signing. While it's a simple concept, this isn't always obtained for several reasons. For anybody who's ever looked at informed consent, you would know that the reading level, they're drafted by a lawyer, so the reading level in the informed consent tends to be relatively high and may be difficult for many to understand. Consent forms and templates are usually written at the 11th grade or higher standard. In fact, in my legal practice, we spend a lot of time looking at informed consents, trying to take out a lot of the legalese and the ambiguities so that a person of average means can understand them and agree to what they're reading or being asked to do. Consent forms tend to be long, but there are also several personal factors that can cause different levels of understanding among different persons. Obviously, when you have a clinical trial that's being conducted, there are a variety of different ages of potential participants. People signing the informed consent might be ill. They might have little familiarity with the research, language differences, might be speaking a foreign language. Literacy could be an issue. The third element of an informed consent is voluntariness. That is, the subject who agrees to participate in a clinical trial must have the ability to make that free choice. There cannot be any coercion or undue influence in making the person sign the informed consent. And here you see in our little cartoon the consent process where the individual is being coerced to sign it. But even without an explicit strong-arm tactic, there could be many other variables that affect voluntariness, including pressure from others, like family or friends who want to see the patient enroll in a clinical trial, trust in a healthcare provider. If you're sick and you depend on your doctor, that physician might have a lot of influence over whether or not you sign informed consent. Restricted choices, maybe there isn't an alternative for your treatment, and so participating in a clinical trial is the best way to address your illness. And incentives, and this is a big issue that we deal with in the legal side, is it's inappropriate to pay doctors to influence patients to participate in clinical trials, but also there are questions about compensating the individuals to participate. It is wrong to pay individuals to take a particular drug for the purpose of conducting a clinical trial. And then the final element is authorization. That is, the person signing the consent has to have the authority to sign it. And this isn't typically an issue unless the person subject to the research is a minor or because of the illness perhaps does not have the ability to understand what he or she is being asked to sign. The next topic that I would like to discuss is should human genes be patentable? And this answer may soon be decided by the Supreme Court. So at the end of this topic 3, if you feel like you don't have an answer, it's because we're still waiting. We don't know what the answer is right now. The Supreme Court agreed to decide this case and whether human genes should be patentable. The case that will be heard by the Supreme Court on this issue is called the Association for Molecular Pathology versus Myriad Genetics. And this press release here just came out in November of this year, so this is very timely. And as I said, it's a very controversial issue which is currently unfolding. And this might be some interesting conversation for a cocktail party, but I'll give you a little bit of background about what this case involves. There's 2 genes in question, the BRCA1 gene and the BRCA2 genes. And anybody familiar with breast cancer has some knowledge of these 2 genes. These genes are used to detect risk of breast and ovarian cancer and aid in treatment options. They're generally very good predictors. Women who test positive using Myriad's gene test and these 2 biomarkers as an indication for the risk of getting breast cancer have an 82% higher risk of actually getting breast cancer. And those who use this test for the detection of ovarian cancer and have had a positive result have a 44% higher risk of ovarian cancer in their lifetimes. So the tests are extremely effective. So what are the 2 sides of this debate? Myriad, who is fighting for the patent protections for genes, argues that denying patent protection could slow advances in medicine. Essentially, if there aren't patent protection, then there's inadequate incentives for biotechs and financiers to develop these types of tests. Others, such as the ACLU, Women's Rights Project, argue that patenting genes is problematic because doing so would prevent many people from getting access to the necessary information that they need so they can understand their genetic risk of disease and they can plan their medical care appropriately. So as you can tell, this case presents one of the most contentious and potentially evasive issues in patent law. The case to be decided by the Supreme Court is, what is the difference between an invention and a natural phenomenon? And can genes, therefore, be patented? The decision is going to have an impact on researchers, biotech companies, and patients. And at this point, we just have to stay tuned to see what happens. The final topic that I want to present today is when is it ethical to treat patients with non-FDA approved drugs? At one time or another, almost everybody that I know has relied on prescription drugs to help treat or cure a symptom or an illness. In fact, approximately 2 billion prescriptions are filled each year in the U.S., but few of us know of the complicated and lengthy process involved in creating and providing a new medication to the public. And this slide shows some of the processes of drug development. The process of drug development can be divided broadly into two categories, preclinical and clinical work. In the preclinical stage, big pharmaceutical companies and biotechs engage in a process to identify new chemical entities for the treatment of disease. These candidates will have promising activity, typically against a particular biological target thought to be important in disease. However, at this point, we will know very little about the safety, the toxicity, the pharmacokinetics, or the metabolism of this drug in humans. Before a drug can be tested in humans, it is important to assess all these parameters. A further major objective of drug development is to make a recommendation of the dose and the schedule to be used before a compound goes into a human. In addition, drug development is required to establish the physiochemical properties of the compound, that is, the chemical makeup, its stability, its solubility. Drug development will also involve developing the process by which the chemical will be optimized, so that when it's at the university and it's being made at the bench on a very small milligram scale by a researcher, it can be scaled up and manufactured on the kilogram and then even on the ton scale by a big manufacturing company. The drug candidate will be further examined for its suitability to be made into various forms. As we all know, those of us who take drugs, there's a variety of different forms a drug can come into. It can be a capsule, or it can be a tablet, it can be an injection, etc. Before a drug can be tested on humans, many regulatory requirements must be satisfied to the satisfaction of the FDA. These generally constitute a number of tests designed to determine the major toxicities of a novel compound prior to its first use in a person. It is a legal requirement that the assessment of major organ toxicity be performed. This is called a tox biodistribution study. Essentially, we look at the effects of the drug on the heart, lungs, brain, kidney, liver, and digestive system. Some of the tests can be done in vitro, meaning we use cells and not living animals, but many of the tests do require experimentation on animals, just because sometimes you really don't know what's going on unless you look at an intact organism. The information gathered from the preclinical testing is submitted to the regulatory authorities, and of course in the U.S., the FDA, and that's submitted as an investigational new drug, or an IND. If the IND is approved, then development moves into the clinical phase. And the clinical phase involves 3 steps. Phase 1 clinical trials, and that usually involves healthy volunteers. In this phase, we determine the safety and dosing of the drug, dosing meaning, how much you should take and when. Phase 2 clinical trials are the next step, and that basically is used to get initial reading of the efficacy and to further explore safety of the drug in a small number of 6 patients. The Phase 3 clinical trial is a large pivotal trial, and this could involve thousands of patients, especially in cardiovascular trials. These are large pivotal trials to determine the safety and efficacy in large number of patients. If a compound emerges from the clinical trials testing with an acceptable toxicity and safety profile, and it can be further demonstrated to have the desired effect in clinical trials, then it gets submitted for marketing approval in the U.S. or in other countries where it will be sold. In the U.S., this process is called the New Drug Application, or the NDA. Most compounds, however, fail during this process of drug development. Either they fail because they have some unacceptable toxicity, or they just don't work. In fact, I saw some statistics the other day. Approximately 70% of drugs that go into Phase 1 clinical trials fail, and even drugs that make it all the way to Phase 3 clinical trials, approximately 30% of those drugs fail. So drug development is very risky. It also takes a very long time to develop a drug. In general, it takes an average of 12 years for a drug to go through that process that I just showed you on the last slide. Only 1 of 5 drugs that we identify in the lab is actually finally approved for human usage, and some numbers I've seen are actually even lower than that, could be as low as 10%. On average, it takes $350 million to develop a drug. If it is a cardiovascular drug, that number increases substantially. I've seen numbers as high as $700 million to develop those drugs. So drug development takes a long time, it's costly, and it's very risky. So if you're a sick patient and you have a disease for which there is no drug currently, and your timeline doesn't look very long, it looks very bad. Your outlook looks very bad if you have to wait for a drug to progress, through the 12-year process. And when you look at the likelihood that the drug is going to be successful, it's relatively low. This could look very demoralizing to you. So what do you do if you're a sick patient and you are looking for some type of treatment? Well, generally patients can only take drugs that have been approved by the FDA after completion of the full drug approval pathway from preclinical testing all the way through the 3 phases of clinical trials and then approval of an NDA. However, if patients do not respond to the drug or are intolerant of approved therapy, there is the possibility that they could get access to investigational drugs. That is, drugs that have not been approved by the FDA. And there's 2 ways that patients can get access to those drugs. First, a patient can enroll in a clinical trial and get access to a drug that's being tested in that clinical trial. Or second, they can get access to a drug through the Expanded Access Program, otherwise known as the Compassionate Use Program. And this allows the use of investigational drugs to treat a sick patient with a serious disease that does not have a satisfactory alternative to treat the disease. The 1997 FDA Modernization Act allows a patient to obtain investigational drug when the patient's doctor determines that the patient has no satisfactory treatment. The FDA determines there's sufficient evidence of safety and effectiveness to support use of the drug, and the drug will not interfere with ongoing clinical trials. The sponsor also has to submit, however, some information to the FDA. Essentially, the information has to be sufficient to satisfy the IND requirements, and so that means that there has been a level of testing of the drug, usually in large animals. As we mentioned previously, this would be, the tox biodistribution studies to show where the drug goes and the effects it would have on various organs. Again, we're looking at the issue of safety more so than efficacy with this level of testing, but hopefully the large animal studies also show that there's a sufficient amount of efficacy such that it justifies the patient taking the drug underneath the compassionate use exemption. The benefits to patients of compassionate use drugs can be significant. I've listed a few of the benefits to patients here on this slide, but most importantly, this offers hope for patients with no available alternatives. But there are several considerations for compassionate use, including, of course, the minimization of risk to the patients, that is, ensuring their safety. For patients that are on the verge of death, this is less of an issue. If they are healthier patients, then it's less likely that an exception will be allowed for them to take a compassionate use drug. Access to investigational drugs must always be balanced against the benefit of the conduct of clinical trials. Clinical trials are the best means to obtain evidence regarding the likelihood of the effectiveness and the safety of the drug in large sample sizes, and the FDA approval is the most efficient way to make a drug widely available. In general, patient autonomy must be balanced against also the potential exploitation of desperate patients. Obviously, a patient is very ill and there's no current treatment available for them. They're more willing to put themselves at risk and it's always important to make sure that those desperate patients are not exploited. In general, the decision to use a compassionate drug is a variety, it's a community decision of a variety of different participants, including the patient, his doctor, the sponsor, that is the biotech and pharmaceutical company that developed the drug, the FDA, and the Institutional Review Board of the IRB, which is responsible for ensuring the safety of a protocol, clinical trial protocol that's to be conducted at a particular hospital. Even for patients who have no other options for treatment, there are considerations. The patient may be overvaluing the potential benefit of the drug and underestimating the risk. The cost of the investigational drug may not be paid by insurance, and this may impose a huge financial burden on the patient. And there is a risk greater than death, surprisingly. New drugs could have toxicities that cause increased suffering and pain, or accelerate, or in some cases, if it's a poor quality of life, prolong a bad state. The decision to use an investigational drug has an impact on doctors. He has additional paperwork that he must complete, and he may be unfamiliar with the process for the use of these drugs. The physician may not be able to recover all of his costs in providing the investigational drug and may face liability issues for the unknown negative side effects of the drug. The decision to use an investigational drug may have an unexpected effect on biotech and pharmaceutical companies that develop them. The companies must be able to provide the drug in sufficient quantities for the patients who need them. There may be cost or capacity constraints. And what do you do if a drug is effective and many patients are clamoring for them? How do you allocate the few numbers of drugs that are available to the multitude of potential patients? The results from the use of the investigational drug may affect the regulatory review and decrease the likelihood of FDA approval. I have faced this issue many times in my legal practice, where a patient will call up a biotech or pharmaceutical company and want access to a drug that looked very promising in a phase 1 clinical trial. And the biotech or pharmaceutical company, by giving a compassionate use drug to a patient, risks that some kind of adverse condition will result. The drug won't perform as well as expected because maybe that patient wasn't appropriate, 100% appropriate for taking the drug, didn't fit the typical inclusion criteria or the profile of the type of patient we would look for as the ideal candidate for this drug. And so as a consequence, there's now some kind of blemish with regards to the effectiveness of that drug that might not have resulted if the patient was screened through typical clinical trial protocols and was more appropriately suited for the profile of what we were looking for with respect to drug treatment. So there are several issues that biotech companies have to look at. In addition, if a compassionate use drug is given to a patient and it doesn't perform as well as expected, this could affect the company's ability to raise capital, and that's always an issue for biotech companies who are trying to advance their therapeutics. But there's also an impact on the FDA. The review of compassionate use drugs requires additional time and effort of the FDA. It's already pretty constrained, and often the review of the request to use a compassionate drug has to be done on a quick turnaround basis because the patient is pretty sick. And so again, it just puts more resource constraints on an already constrained institution. It has an impact on the Institutional Review Board of the IRB. The IRB is tasked to make sure that the clinical trial to be conducted at a particular hospital is safe for the patients. The IRB, in order to make this determination, has to use additional resources to review the treatment. The IRB might not be familiar, many times they're not, with the compassionate use process and what's required by the FDA in order to advance that, the use of that drug at their institution. So there's many issues with regards to the use of investigational drugs. And a few lingering thoughts just for future consideration is who pays for these drugs. If the manufacturers, then there's a disincentive for manufacturers to develop drugs. If insurance companies, experimental drugs are generally not covered. And if the patients have to pay out of their own pocket, then there's the negative consequence that perhaps only the affluent will receive access. And finally, as an overarching consideration, we also have to be concerned with how do we reduce the risk of exploitation and fraud for this very vulnerable population that perceives it has no alternatives for treatment. And so with that, I have presented 4 topics for consideration. Hopefully these have created some thought-provoking questions, and in the last few minutes, I'll take any questions that you might have.
Thank you, Sheila. That was a great presentation with a lot of important information. I have posted the questions up on the screen, and I'm turning that back over to you to go ahead and address the few questions that we have.
Has Henrietta, so the first question is, has Henrietta since been compensated for the HELA cell use? And the answer is no. Her family has received no compensation. In fact, there was a book written about this that's actually quite good. In the book, what is it, The Immortal Life, I think, of Henrietta Letts. I think that's what it's called. I read it probably about 2 years ago. The person who wrote the book, the author, interviewed the family members, and as you can imagine, they're quite angry and resentful that they haven't received any compensation. They are not financially well off and don't have the means to pay for health care, and so obviously having some of the, sharing in some of the financial returns that the manufacturers of HELA cells have, been able to obtain over the years would have been very, very useful for that family. But no, there has been no legal judgment, and there's been no compensation to Henrietta Lett's family for the use of the HELA cells. So that's unfortunately the answer to question number 1. Oh, please give an example of selling tissue for art, stated in slide 11. Well, that's interesting. I have to say I haven't gone to too many art galleries that have tissue displays, although in my office I do have many art posters hanging up that show different kinds of tissues. So, there's this whole explosion of art that uses different kinds of cells and tissues, and the staining of those cells and tissues, and just fabulous. It creates fabulous visions, and it's almost like futuristic art. So I have many of those, and I'm actually looking at one right now as I'm speaking to the audience, hanging in my office, where people have used tissue to create photos and different kinds of art hangings that I know are very popular. But other than that, I can't say I know of too many people who have actual tissue hanging up as art. Probably the only thing that I can think of that even comes close is here at South Point a few years ago, they had this exhibit of human bodies, and maybe that's the closest thing that I can think of where there was selling tissue for art, although arguably that was somewhat for educational purposes too. But that's probably as close as I can get. The third question is, how do you handle objections from people opposed to animal testing and even human testing in clinical trials from a humane perspective? That's a really good question. I'm an absolute animal lover, and so I struggle with this all the time. One of the companies with which I worked and had co-founded was working on a treatment for muscular dystrophy. And unfortunately, the animal model for muscular dystrophy is a special form of golden retriever. These golden retrievers are bred to have a dog form of muscular dystrophy, but essentially before you can go into any humans, you have to test the therapeutic in these poor dogs. I'm an absolute dog lover. I have a golden retriever who comes to work with me most days and is my my lifelong companion, his lifelong at least companion. We're always together. And so I can understand the issues related to animal testing. And, you know, it's a horrible sacrifice. It really is, that we have to make of these animals. But the bottom line is we just do not know how drugs are going to perform before they go into humans, and we need some means to do so. Using a dog, even a dog that I love, such as my dog Argos, is much more humane than using, in my case, for muscular dystrophy, somebody's child. We don't want to put muscular dystrophy, for those of you who don't know, is an illness that affects young boys. Typically, they stop walking by the time they're 6 or 7 years old, and by the time they're in their late teens, they usually pass away. I would much prefer, and I always tell people this who have objections, to test this therapeutic, which could have great risk, in a beloved dog, right? Which I really do love animals, before we test it in, you know, our children. So it's unfortunately just like, you know, those people who go off to war, they make great sacrifices, and I think the animals, we have to acknowledge the great sacrifices that they make, but it's just essential because there's no way we know how drugs are going to perform, despite all of the cell, you know, testing we do until we get it into an animal, and the animal testing models are very important to do that. So that's that question. You know, there's no really good answer. It's just, unfortunately, sort of a half-fulfilled answer, but that's the best I can do. And number 4, why is there not a fast-track process effort for developing, patenting, and approving genes or any biotech product to speed up treatments or cures? Or will we always be subject to the same slow process for drug approvals? This is a great question, and this is something that we in the biotech space struggle with all the time. It costs so much money. It takes so much time. It's so risky, and it's just, you know, it's very hard these days to get money for early stage technologies, because when investors are looking at where they can put their funds, it's, you know, much more assured to put it in the stock market and Procter & Gamble stock in many ways than in a biotech company, just because of the risk and the time before you get back your money. But why I get so frustrated with it, and I can totally sympathize with this question. We also know that there are so many risks with drug development, even when we go through the process of a phase 1, phase 2, and phase 3 clinical trial. As we learned from Vioxx, which was a drug to treat heart disease, there can be very negative consequences associated with these drugs. And so it's a fine line, it's a balancing act, trying to make sure we get enough information about the safety of these drugs before we approve them for wide use, and the cost and time, right, to get these drugs approved. It's frustrating, I think, for the FDA as well as those of us on the other side trying to push these drugs through. It's a very hard process, and unfortunately, I think we need to learn more. If we can know more about how drugs are going to perform before we get into human use, I think that's going to expedite the development timelines. And I get really excited by a lot of the modeling we're doing now with computer simulation, and I'm hopeful that that will give us an expedited path to get these drugs approved more quickly. And maybe going back to question number 3, if we can have better models that predict how drugs are going to perform before they go into humans, we might even be able to decrease the amount of, you know, animal testing that we do. So great questions, and I appreciate, you know, those thought-provoking issues. So great, I don't see any more questions, and I think we're like out of time, like right on time. Is there anything else that I should address at this point? Oh, there's more questions.
Yes, Sheila,
they just popped up.
They
just popped up. Okay, great. So, okay, as everybody can tell from this this presentation, my computer skills are not the best. This is why I went into biotech, because I could not do software or information technology. Question number 5. Are any biological discoveries found from these excruciating historical experiments? Unfortunately, yes, or maybe fortunately, yes, depending on how you look at that. I don't want to say that the means justify the ends, but even the Nazi experiments gave us some great insights to how the human body performs. And that has been instrumental in, advancing science. So I really have to be careful here because, of course, right, those experiments were heinous and totally unacceptable. But we have learned from some of these situations that I took from history, we have learned things which I guess in some ways, at least for those patients who are victims of those experiments, we have benefited. The syphilis trials gave us some information about how that disease progressed, which was important for the time. But again, right, I don't want to say that the means justified, or the ends justified the means in any way. Number 6, aren't most waivers of rights to cell lines incorporated releases for normal treatment procedures? Yes, actually they are. They are commonly incorporated into releases for normal treatment procedures. So this person who raised this question is very astute, obviously reads what he or she is signing. It is commonly incorporated into the releases. And so today when you go to a medical institution and undergo any type of routine treatment, you most likely are waiving your right to cell lines. Still a lot of ethical questions if that cell line subsequently generates, is developed into or if those cells are developed into a cell line that generate, millions or billions of dollars. Shouldn't the patient who contributed those cells share in the financial remuneration? The question for debate, as we discussed earlier in this presentation. But no, that is correct. Number 7, because of U.S. restrictions, is this why people seek alternative treatment in other countries? What is being done to address this instead of just telling people not to? That's an interesting question. Because sometimes a drug takes longer to get approval here in the U.S., people do seek alternative treatment in other countries, and there have been instances where the drug could be approved in other countries and still not approved here, and people go to outside the U.S. to receive access to that treatment. It's a fine line. The FDA and the biotech company, in many instances, want to make sure that the drug is safe. The U.S. standards are often some of the highest standards, if not the highest standards in the world with regards to drug approval. But I helped a company and a patient last summer, and people, are flexible. I had a patient who knew that I was working with a company on the West Coast that had developed a treatment for cancer that was in the experimental clinical development, early phase 1, 2 clinical stages. This patient had been basically given a death sentence, really wanted access to that drug because there was no alternative available. Got in contact with the biotech company. The biotech company gave access to that drug, and amazingly, when was it? On January, I remember this well, on January 4th, it was my 25th wedding anniversary. He called me up at 6:30 in the morning saying he had found out that he just got back test results and his cancer was in complete remission, which is a phenomenal story and makes anybody in the biotech space feel really good about what we do. But the bottom line is everybody tries to work together, the FDA, the biotech companies, patient advocacy groups, to make sure that if there is a situation where somebody desperately needs access to the drug, that we do everything possible to get that person access to the drug. But it's always trying to balance the risk, the safety risk, and the potential benefits, and it is very tough. It's extremely tough situation, and there's a lot of reform occurring to try to make it easier to get access to these drugs. How are healthy persons recruited and selected for phase 1 clinical trials? What is the carrot? This is a really great question. I'm currently facing the situation for a clinical trial, phase 1 clinical trial, for a gene therapy treatment for hemophilia B. Hemophilia is a bleeding disorder. Gene therapy is perceived as a little bit more risky. So how do we get, this isn't a healthy person necessarily, this is a person who is responding well to current medications. So in that sense is healthy. How do we get that person to enroll in to get off his current drugs that are working and enroll in an experimental drug? It's really hard. These are special people that do this, to be quite honest. They are people who often aren't satisfied with their current treatments. Maybe in this case, injecting a drug several times a week is problematic, doesn't make them feel good, it's painful. And it's also people who want to make an impact in society and in their patient population. They want to make contributions. So these are truly special people. We cannot, in the U.S., pay them large, dollars of money to participate in these drug trials. So this is really a group who's doing this out of a voluntary nature and who are, trying to do something that contributes to the advancement of science. I mean, they're just very special people. So I think that's all the questions at this point. Greg, unless you pop up some more.
That's all we have. I actually had to have people hold their questions because we were getting so many coming in fast and furious at the end here. This is a little bit different than I normally close and wrap up today. I'd like to put Sheila's email address up here because there have been several folks who have privately messaged me and asked me if your PowerPoint presentation would be available for them to get a hold of later, and I'm just going to have you address that on an individual basis. However, I will note that today's presentation was recorded, so it will be visible again. Everyone who participated today will receive a thank you email from the BioNetwork, and that will include a web link to this recording. It's not a PowerPoint in the traditional sense that you'll be able to show them in a classroom, but you'll be able to show this entire presentation in the classroom with the audio. So that actually makes it a little bit better, in my opinion. But if it's okay with you, Sheila, I'd just like to put this up here so that people know how to reach you, and
no problem.
If they have any further questions, they can ask you those questions. I would also like to thank everybody for their attendance today, and as we wrap up, I want you to take a moment as you log off to answer a brief survey that will appear. Your feedback helps us to plan future BioForum events, but be sure you click submit when you're finished. Sheila, I'd like to thank you for being our panelist today and doing a wonderful presentation. We have really enjoyed that, and I would like to remind everybody about our May 7th BioForum coming up entitled Blended Learning, and that will be May 7th at 10 a.m. And most everybody in attendance today will get that invitation in an email from me in the next several weeks. Thank you again, Sheila, and good luck to you, and I really do appreciate your taking the time out of your busy schedule to help us out today and teach us something that has been asked for us to present for a very long time in the BioNetwork. On behalf of the BioNetwork and all of us in the state of North Carolina with the community college system, we thank all of you for your attendance and hope you have a great day. Thank you.